It's my pleasure to introduce NanoVation Therapeutics. My name is Jayesh Kulkarni. I'm the CSO and co-founder at NanoVation. Before I get into the details about our technology, I just wanted to provide a brief intro to who we are. We're a platform company headquartered in Vancouver, BC. Chairman of our board is Peter Cullis, generally referred to as the founding father of LNP technology. Dom Witzigmann, our CEO. Early on in our existence, we also hired in Wendy Lampson as their chief IP officer, and you'll understand the specifics of that in just a moment. Don Enns is our chief business officer, who is formerly the CEO at Northern Lipids and then VP head of global drug delivery at Evonik. We also engage with Molly Stevens in the UK, now at Oxford, as a key opinion leader in the nano space. And one of our key founding members also is Dr. Marco Cipollini, who was quite instrumental in the design of molecules such as KC2, and that eventually led to structures such as MC3, which are lipids used in commercial formulations. And our goal really is to bring together the right set of expertise to help generate the next generation of nucleic acid therapeutics, improving on technologies that currently exist. And just to give you a flavor of what that could look like, a quote from Peter in Nature Reviews Materials that says, almost any disease you care to name can potentially be treated using an RNA-based gene therapy. And so, there's an immense potential in RNA-based gene therapy, but of course there's critical limitations in the delivery and how that's enacted. In the image that I show on the left, the first ever RNA interference product called Onpattro was a lipid nanoparticle that encapsulated siRNA and delivered to hepatocytes in the liver following intravenous administration. It largely formed the basis for current LNP technology. And what we've seen with the mRNA vaccines, for example, transition of to newer lipids that really help to boost the potency and really, incredible achievements with modifying the system to provide durable and robust immune response. However, both of these systems, when they're intravenously administered, still accumulate in the liver. And so one of the key pieces to address was, if you wanted to get outside of the liver, you had to re-engineer the system to be able to get to those, get to other tissues as well. And so what we've focused on within NanoVation is to try and address this particular need. In addition to a more, let's say, business or let's say a technological limitation, and in particular, the current approach that's typically used in order to commercialize an LNP or a lipid nanoparticle RNA formulation is that developers have to access technology from a number of different suppliers, and this can add up relatively quickly. It's a pretty complex process, easily done, of course, in the context of pandemic, but now that we've moved on from that, the ability to do this in a more effective manner is not as straightforward. And so what we're building within NanoVation is a toolbox that contains a number of these different pieces, and we break the technology down really into four main pillars, or four drawers, if you'd like. The specialty ionizable lipids, these are molecules that are effectively what help to promote encapsulation of nucleic acids and also intracellular delivery. And so designing these with safety and efficacy in mind, so thinking about how they turn over once they've done their job. The second piece is on LNP composition. So when you combine 2 or 3 or 4 different lipids, what are the ratios in which those lipids are combined, and how that can be leveraged in order to enable delivery to extrahepatic non-liver tissues. Surface modification. So how do you, during the pandemic, the polyethylene glycol lipid component received quite a bad rap, and whether or not that's justified is a different conversation, but generally speaking, these surface modifications were intended to improve the stability of the particle and in turn have received a sort of negative view. But our perspective on this is, if there's a fourth component that's to be added and needs to perform that function, can it also be leveraged for functional elements rather than stability alone? And so improving both the tolerability and targeting of these delivery systems, leveraging that fourth component. And we do have earlier and more research-based efforts on RNA modifications. The idea here being, how do you manipulate the chemistry of the RNA in order to increase the amount of protein that comes out of each RNA molecule? And all in, we're hoping to build NanoVation into a one-stop shop IP portfolio to support commercialization of our partners. And almost everything that we do, that we look to partner out on, we have a very strong focus on the patentability of the technology and the ability to commercialize. Freedom to operate is a critical consideration. Just to give you a quick flavor of the types of technologies that we have within our portfolio. The first one that we call the LT-LNP, which is the long circulating LNP, is really the basis for how we access extrahepatic tissues. The traditional on-patch composition or 50-10 composition, when imaged under cryo-electron microscopy, looks like this colored in circle. We've developed a new formulation that has extended circulation profiles and displays a dramatically different morphology, where you see the sort of clear bilayer structure and a overall different morphology. When intravenously administered, these formulations also display different biodistribution and clearance profiles. So when we look at the image in the middle here of a SPECT-CT demonstrating where the particles end up, we see with the typical LNP, you hit the liver quite clearly. With the LC-LNP, though, there's dramatically less that accumulates in the liver. The spleen lights up. At this particular time point, the heart lights up, but that's not because of true delivery to the heart. It's that there's still a considerable amount left in circulation. And when assessed using a radioactive label that doesn't exchange from the particle, what we demonstrate is that circulation time goes from, instead of 15 to 30 minutes, out to 2 to 4 hours. And we've now even developed formulations that go out to 8 hours. And this extended circulation is really what enables delivery to extrahepatic tissues. And we've focused on not using things like cationic lipids, anionic lipids, molecules that are generally considered potentially toxic or challenging to work with downstream, but rather just focused on really modifying the ratios of the components so you can really make this LC-LNP using the exact same 4 components that are in a clinically approved formulation. And that composition is really what drives the distribution and clearance. The impact of working on these sorts of formulations is demonstrated here. So on this slide, I show the impact of leveraging both our LC-LNP technology combined with our own lipids. First, the LC-LNP compared to a benchmark. When we deliver luciferase mRNA and we look at the activity in the bone marrow, we see a dramatically improved activity with the LC-LNP compared to the traditional system. But then once we start to mix and match that with our own ionizable lipid library, we've started to see, up to a 5-fold improvement in activity in bone marrow. And so this sort of mix and match approach is what we've leveraged to really design next-gen therapeutics. And I just want to clarify on this point that as a result of this mix and match approach, it's not that we get 100% of the delivered dose to the bone marrow or anything like that, but we do see improved delivery to the bone marrow. We see improved activity in the bone marrow. We do see some residual activity in other tissues, but manipulating the cargo in that context has worked quite well to improve tissue specificity. And so in terms of how we've partnered and worked with different groups, we're a platform company, and we're not in the position to vertically integrate. We're not looking to do that. Our goal is to really focus on our sweet spot, which is this early stage prototype development, where we bring synthetic chemistry expertise and mRNA and formulation together, assess the activities in early preclinical models, and then work together with partners to drive that forward. And so our three main goals for BioUS this year are looking to meet with potential partners and biotech pharma interested in developing these nucleic acid therapeutics. We're looking to meet with CDMOs for downstream scale up and manufacturing considerations. Our focus here really is on fully integrated CDMOs. And then finally, meeting with regulatory groups that can help us direct our regulatory affairs efforts. So with that, I'd like to thank you all for listening and the opportunity to present today.